Assalamualaikum warahmatullahi wabarakatuh, and a very good afternoon. I bid to Madam Chairperson, Puan Hana, lecturers and colleagues. So I'm Hariz today, and I will be presenting the project entitled Antibacterial Assessment. So it's quite a long topic, however I can summarize it later. So it is an antibacterial assessment of in situ synthesized bio-inspired silver nanoparticles using recycle chain, or we call it scientifically as aristatus, leaf extract immobilized on the synthesized zeolite, which have been heard commonly. So firstly, I would like to introduce what is actually going on in my project. So basically, I would like to emphasize the novelty of this method by immobilizing silver nanoparticles on the zeolite framework that has been synthesized. So in this project also, we will be using the greener method innovated from the previously used harmful chemicals and energy-consuming physical methods. And also in this project, we will be using plant metabolites contain reducing agent to reduce silver ions to silver nanoparticles. And this whole project will be applied of the materials to know whether the effectivity of the antibacterial of the samples towards bacteria of gram-positive and gram-negative, because we know silver, as in silver ion or silver nanoparticles, are very effective in killing bacteria. The overall overview of my methodology is like this. If you can understand it well, then I have delivered my presentation well. So basically, by immobilizing the silver nitrate into the zeolite, we will get silver ion, or we can get silver Ag zeolite, Ag synthesized, immobilized in the synthesized zeolite. And the process of immobilizing Ag ion is by the ion exchange of the Ag plus with the Na plus. So by immobilization, we will obtain the Ag plus zeolite A. And then after we introduce the plant extract of recycle chain, which we believe has a very rich antioxidant and has a reducing agent, which we call Sina, one of the plant metabolites. This reducing agent will reduce the Ag ion in situ, meaning on site, in the framework of zeolite, and reduce the Ag ion into Ag nanoparticles. In the work of reducing of the Ag ion, the plant extract or the reducing agent also can cap the silver nanoparticles and stabilize it in the zeolite framework. And finally, we will have the bio-inspired, we inspired from the green of nature, immobilized on zeolite A. So I will just only take several results from my study. So to make it very clear, we will just want to see whether the silver nanoparticles are actually immobilized on the zeolite framework. So we have here synthesized zeolite, Ag plus zeolite, the AgNP immobilized zeolite, the third layer, and lastly the commercial PVP cap AgNP. So we can see that there are several peaks observed that is very similar to the commercial silver nanoparticles, which I can see that there is AgNP produced after successfully we synthesize the bio-inspired AgNP immobilized on zeolite. Next is the scanning electron microscopy, or we call it as FESEM or SEM. So actually this is the morphology of the synthesized zeolite, and then after the introduction of Ag ion, actually there is no very significant changes of the structure. It still have its cubic or hexagonal shape, or there's similar edges of the zeolite framework. And then after the introduction of the plant extract into the Ag plus synthesized zeolite, we can see that actually the morphology of the zeolite itself becomes very significant compared to the Ag plus zeolite, which has a very cubic shape over here. You can see that here. So it means that maybe the use of plant extract itself can really help in maintaining, or basically to be exact, stabilizing the framework of the zeolite itself and also the AgNP. Next is the ETM. So this is what we sought for. Basically, when we want to see nanoparticles, because actually we cannot see nanoparticles using FESEM, so we have to use TEM. So we can go deeper into the framework. As you can see there in the synthesized zeolite here, there is no actually nanoparticle. Should be no nanoparticles at all because it's only a zeolite itself. And moving on, when we add silver nitrate as in the form of Ag, there's actually a deposition or immobilization of a very fine and small particles, and it is zeolite. They are silver. Sorry. And then when we add plant extract further, the immobilization of the AgNP itself also in the framework, but by morphologically looking at this TEM, you can see that the Ag is deposited on the surface of zeolite. However, in the form of AgNP, the deposition or immobilization of the silver is inside the framework of zeolite, and that is very interesting. And finally, we would like to see whether the so-called silver can kill bacteria, and they are. So we use the silver or the synthesized samples against E. coli and also Staphylococcus aureus. As we can see, synthesized zeolite without any antibacterial agent, and there is no inhibition zone. Moving on to the Ag synthesized zeolite, there is a very significant large inhibition zone in compared to the silver nanoparticle synthesized zeolite. This may be due to the synthesized zeolite act as the carrier, and the silver nanoparticle itself is slowly released into the media or into its outer environment of the zeolite. So basically, all in all, we can say that silver in synthesized zeolite definitely can kill E. coli and Staphylococcus aureus. And I would like to conclude that this mosaic chain is a very potential herb to be used in greener nanotechnology advancement, not only mosaic chain, but other type of local herbs as well. And the in situ synthesized AgNP also showed commonly less than 100 nanometer, which is definitely nanoparticles. And also the immobilization of silver nanoparticle demonstrates a notable antibacterial activity against these two types of bacteria using this diffusion method, which the physical interaction occurred between the bacteria and the antibacterial agent in the media. And most importantly, this greener approach is as effective as the conventional physical and chemical method. So thank you to UTM and to Ministry of Education Malaysia. With that, thank you.